In order to accelerate and lead others to another day of captivating session, it is my privilege to introduce today's guest speaker, Subramaniam Nateshan, Professor, Department of Pharmaceutics, NIPA, Kolkata. Sir has postdoctorate in pharmacy, Jadavpur University, Kolkata. Sir has done his specialization in industrial pharmacy, Annamalai University, Chidambaram, India. He is graduated from Dr. MGR Medical University, Chennai. Also, Sir has diploma in chemical process, industrial instrumentation and control, Annamalai University, Chidambaram, India. So, Sir has various national and international publications. Sir has patentable works renowned with US, PCT and Indian publications. He has provisional applications on method and composition for solubilization of hydrophilic compounds in India. He has been a principal investigator since 2008 to 2016. Glimpse of his works on novel strategies includes for the improvement of treatment of breast cancer, efficient intraocular delivery of drugs, and drug delivery and detoxifications. So, from the year of 2010 to 2013, Sir has been renowned as a co-investigator for pharmaceutical and allied industries, New Delhi. So, enriching with teaching fraternity, Sir worked at Anna University of Technology, Bharati Darshan Institute of Technology, Tiruchirappalli. And Sir has been worked as a lecturer in Vrindavan Institute of Technology, Pallavan Pharmacies, and then since 1994 to currently, he's been working in Vrindavan Institute of Technology, Tiruchirappalli. Sir has received Best Innovation Award. He has received Boycast Fellowship, Young Scientist Fellowship Award, and Research Fellowship Award associated with Fellowship of Indian Institute of Chemists, Kolkata, India. And for gracing this occasion, now I request our honorable guest speaker to continue with his guest lecture.
Good morning to all. Hope you are able to see my slide.
Yes, sir.
Yeah.
Yes, sir.
Yeah. First of all, I like to thank the Sri Vishnu College of Pharmacy for providing an opportunity to interact with budding pharmacists as well as learned faculty members in the profession of pharmacy. So today I'll be going to give some enlightening points in that area of light-mediated drug delivery systems. As we all know that currently the stimuli responsive drug delivery systems are gaining importance since the delivery of the drug into a particular organ or to the disease site is very, very important. particularly in case of a cancer. The drug delivered into that cancer tissues will also expose to a normal tissues or a normal cells, which will produce some toxic effect. Instead of giving an expected therapeutic effect, we'll also get it a large amount of side effects, which will hamper the treatment schedules, as well as which will alter that lifestyle of that cancerous patient. In order to avoid that, and in order to improve the treatment efficacy, large varieties of approaches has been tried by that pharmaceutical scientists world over. Still, there are a lot of research which is undertaken to target the drug into a specific cancer tissue, cells, area, but that most of the cases will be failed, because the cancerous tissues is also making and changes according to an environment. The reaching the cancerous tissues or that cell is becoming a critical task. Compared to earlier, now we know a lot of new things, how these cancerous cells are behaving, or what are and target, which protein is overexpressed, which receptor is overexpressed, which gene is getting upregulated, which gene is getting downregulated on that specific cancer types. But that also varies in a geographically, which limits that overall targeting strategies for that cancerous tissues. That is, if you take that, in such cases, the stimuli responsive drug delivery systems are becoming an important one. In order to have that stimuli responsive systems, that should be have some smartness. So we'll call it as a some smart system, which nothing but the system that can alter its property in responsive to environmental changes. This concepts has been arrived or learned by the scientists from that nature. Since that nature has a lot of points to learn, to teach us, from which the people are developing a lot of technologies. Or we are trying to mimic that nature and trying to get it in systems which will be suitable for that treatment. So one such example is chameleon, which displays smartness in changing its skin color to blend with that environment. Depending upon that place where that which will sit, chameleon sits, it will change its skin color. So that means depending upon that environment, it's able to change its color. That sensing is very, very important. As like the another concept, we see know that birds, water birds, that able to float in water, due to an oily coating on their feathers, which will provide a buoyancy effect on that for them. Similarly, in a biological systems, every cell will exhibit a smartness. We know that immune system, once that antigen, any kind of an antigen enters into our body, immediately which will be responded by an antibodies or the secretion of an antibodies. Again, our regular thing, that amount of glucose in blood. Depending upon that, that amount of insulin will be produced by an beta cells in an islets of an Langerhans. That is that smartness which has been inbuilt in our system. Now, the people are trying to mimic this smartness in order to achieve an temporal as well as an spatial delivery of a drug into the tissues, for which the stimuli responsive drug delivery systems can be utilized, which has been at a nanoscale level. If I tell that a stimuli responsive drug delivery system is nothing but a system which displays rapid physiochemical transition in response to small changes in surrounding environment. According to that environment, it will changes that, or there will be a physical or a chemical changes occurs in that system accordingly, which will able to release the drug. This system basically consists of three major functions. One is to sense the signal, that is called as a sensor function. Then judge that magnitude of a signal, how much amount of a signal has been produced, that is called as a processor function. Then according to that magnitude, release or exhibit that responses, that is an effector function. So that stimuli responsive drug delivery system should be able to perform these three functions such as a sensor, processor, and effector function. As we know that there are different endogenous stimuli and exogenous stimuli which can be used to activate or to control that drug delivery systems. These systems will be applied into that patient, particularly which will reach us into the disease target, or based on this disease target or the pathological conditions of this disease target, there will be a certain exogenous stimuli, endogenous stimuli, which will be present in the body. Either it may get decreased or get increased. Say for an example, in cancer cells, we say that the pH of the cancer cells will be less compared to this and adjacent areas. Or we can say that there is a higher amount of an redox enzyme is present in the cancerous cells. So accordingly, that endogenous stimuli can be considered, that drug delivery system can be designed to deliver that required amount of drugs. That is that another big area that currently the scientists are targeting is an exogenous stimuli, where the temperature, magnetic system, aquatic systems, or light may be utilized. So today's our talk will be respected to these light-mediated drug delivery systems. If we consider all these stimuli responsive drug delivery systems, including light-activated, will be able to control as well as release based on release of the drug based on that demand. It may undergo some reversible changes in that nanostructures, switching off between an on-off mechanism, as well as, if need, which can be able to release us that multiple dose of the drug at various time points, in order which will also can be utilized to achieve the therapeutic range within that single dose. That will be decided depending upon that pathological conditions and necessity of that drug substances, which can also be able to localize the drug delivery into into a particular organ, tissues, or into that cell. In turn, that suitable drug delivery system will be able to preserve or make that stabilizes that load, drug load. In turn, which will be able to offer an improved patient compliance. Various stimuli responsive nanocarriers can be utilized for these purposes. It starts from liposomes, nanoparticles, nanovapors, hydrogels, nanocrystals, nanomicelles, trentimers, implants, microparticles. We can put it up on large number of variations in that lipid-based systems also, which will be stimulated based on that stimuli, which will be able to deliver the drug into that site. So I'll come to that specific where we will be concentrating today's talk with respect to an light-mediated drug delivery systems. In 1903, Niels Reiberg's Fenton has earned a Nobel Prize in medicine for inventing and treatment of an lupus vulgaris by using that ultraviolet light. That is the starting point where the people started to use light for the treatment purpose. As we know that light, which is widely used for the diagnostic purpose, or some of that device where we will be also using a light in combination with the diagnosis and treatment, which is called as a diagnostic systems. But after that Niels invention, the people concentrate lot of things in that drug delivery systems. The light, particularly applying a tool for an on-demand drug delivery due to its non-invasive nature, as well as an ease of application and ability to control both in that spatial as well as temporal regions. Currently, there is a lot of development has happened in this area due to the technology development in that nano sciences or nanotechnologies, as well as that a lot of research is going on nanomaterials or fabrication of nanomaterials with different physicochemical properties. So the advanced nanomaterials such as metal nanoparticles, metal sulfides, oxides, or metal organic frameworks, carbon nanomaterials, upconversion nanoparticles, semiconductors, stimuli responsive micelles, polymers, liposomes based nanoparticles are applied or utilized for this light mediated drug delivery or light stimulated drug delivery. So due to increase in this demand in this field, a lot of focus has been currently paid over here, but still it has certain limitations. So today's we'll go through that, some of an case studies as well as that limitations of that systems. The primary challenge in these clinical applications of this light mediated drug delivery is poor penetration depth of that low energy electromagnetic radiations. As you know that the different light source which can be utilized for that light mediated activity systems, wherein that light sources UV visible light will show us a minimum penetration depth compared to an X-ray and radio waves. So the low energy near IR region will be able to penetrate up to 5 mm below that skin surface, whereas that light of an high energy of an 200 to an 400 or an up to an 800 also will be sometimes less than that of an few mm, very few mm depth which can go inside. So based on that available data, the optical window for a photo regulation of that drug delivery systems or for an prognostics, as well as for that penetrating wavelength will be and visible to IR region will be of an 650 to 900. Now will be people are going with an 2 photon which has been activated by near IR region, second near IR region, where that more penetrations will be possible. Now we'll come into that categories of a light-mediated drug delivery system, which can be divided into three categories. One is an photochemical therapy, another one is an photothermal, and then photoisomerization. Or we can say that photothermal responsive, photodynamic therapies, or an photo conversion therapy, drug delivery system. The photothermal, we can say that photothermal responsive drug delivery systems. The photochemical can also be called as a photodynamic drug delivery system, that photoisomerization, or we can say that photoconversion drug delivery systems. Either way, which can be classified based on a therapy or a drug delivery systems. Our discussion will be based on a drug delivery systems. The first case we will be see that what is a photothermal responsive drug delivery. The photothermal responsive drug delivery achieve that on-demand release of the drugs via nanomaterial mediated photothermal therapy under light activation. Particularly, our discussion will be held with near IR laser irradiation, which in turn generate that heat to destroy that thermal responsive materials. That's basically, you have that material, to that material will be passing that light will be light energy will be converted into a thermal energy. That thermal energy will kill the cancerous cells. That is a basic concept over here. We call it as a PCM, that is a phase change materials, photothermal responsive phase change materials, which can be exposed into that near IR radiation, laser radiation. Due to that phase changing, there will be a release of the protons, which in turn generate that heat inside that cellular system. That generation of the heat will be able to kill that cancerous cells. Due to that excellent photothermal conversion efficiency, that near NIR absorbing dyes, polydopamine, Prussian blue, copper sulfide, bismuth sulfide, and gold nanoparticles has been widely used as materials for photothermal responsive drug delivery systems, which may be in two types. One is photothermal responsive liposomes or an PCM based halo nanostructures. That next case, photodynamic responsive or photochemical, wherein we'll say that photodynamic responsive rely on that photosensitizer mediated photodynamic therapy, wherein we'll use some light source to activate that photosensitizer. That activated photosensitizer will be able to generate that reactive oxygen species and in turn induces an cancerous death. This generation of an reactive oxygen species will also utilize that oxygen available in that cell. In turn, which will create that hypoxic conditions within the tumor region, which can be utilized for hypoxia sensitive drug delivery systems. So either photodynamics as well as that induced hypoxia or that hypoxia conditions of cancerous cells can also be utilized for stimuli responsive systems. Or in most of the cases, the both at light as well as hypoxia conditions can be utilized in combination to enhance that therapeutic effect. The common photosensitizer for PDT includes an tetraphenylchlorine, indocyanine green, rose bengal, chlorine E6, porphyrin, boron dipyromethane, generally called as a sand body, conjugated polymers or semiconducting polymers, which can be utilized as a sun photosensitizer. There are for an ocular use, photosensitizer called vertifrifin has been already approved for photothermal therapy, so that indocyanine green has been already approved by that US FDA, both for diagnosis as well as treatment. The next one is photoconversion responsive drug delivery systems or photoisomerizations. We can put it some of technologies where that drug delivery systems are fabricated through integrating UV visible light sensitive components such as 2-nitrobenzyl pyrophyrin, coumarin, 2-nitroindronil, donor acceptor stent house, and the most widely used component is an azobenzene into an upconverting nanosystems with loadings as well as conjugation of the drugs. There is another major component which is used over here, that photoconversion system is upconverting nanoparticles, which is mostly based on an lanthanide like erbium, yttrium, samarium, like that. So under near IR laser radiation, that upconversion material convert and near NIR light into an UV or an visible light. That is a photoconversion responsive system. There is an upconversion nanoparticle which is exposed into one near NIR region, which will convert into an UV visible or visible light, ultraviolet or an visible light that can destroy that sensitive moieties, or that will alter that polymeric membrane or break that membrane, which in turn releases that drug. That is one way. Or this light may activate that photosensitizer. That photosensitizer releases that nascent oxygen or reactive oxygen species that might induce us an cancerous death, cell death. The first thing we'll discuss about photothermal responsive systems, wherein we'll take it as two cases. One is photothermal responsive liposomes. There are a lot of studies has been done. I'll be taking one case studies to give an ideas for that audience. Herein, in case of photothermal responsive liposome, the concept is like that. The liposomes will be activated due to light, which will consist of phase changing PCM, phase changing material, that will be responsible for generating heat or thermal energy within the systems. So this system will consist of an eutectic mixtures of a natural fatty acids as well as an chemotherapeutic drugs, that for an example doxorubicin, as well as near NAR dye, which is an IR 780 for NAR triggered release. So the biocompatible fatty acid eutectic mixtures will be able to melt at 39 degrees Celsius has been constructed using an lactic acid and stearic acid. That IR 780 served as a photothermal agent to increases the temperature under 808 nanometer IR laser irradiation, which will induces that melt of an liposomes for the drug release. So this system has shown a cell death of an human lung cancer cells, that is an A549, after the treatment with dogs IR 780 drug delivery systems upon that near NAR radiation reach up to 90%, as well as which is able to show and 1.3-fold higher efficacy compared to one with and without light sensitive systems. There is another system which is an degradable photothermal responsive liposomes, which has been manufactured or produced by using of an cyclic arginine glycine aspartic acid. Simply we call this as an RGD conjugated DSPE as well as an TPPC, that is an polystyrene polyethanolamine or an dipolymmethyl polyphosphatidyl choline, cholesterol and photosensitizer of an ICG indocyanogreen and polyamidoamine, that tendrimers grafting cisplatin prodrugs. So that prodrugs, which is made up with that cisplatin with this membrane, this polyamine pumps. Then this photosensitizer was loaded in that outer layer of FAM PT, which is encapsulated inside that liposomes. So in all together, we can say that liposomes made up of phospholipids will be covered by an tendimeric systems as well as the consist of an prodrugs. That layer will also be consist of an photosensitizer. So these systems have showed better circulation time as well as better targeting capability due to the presence of an RGD. So which will be able to target that RGD's receptors present on the cancerous cells. Under IR radiation at an 808 nanometers, that ICG raised that local temperature, which in turn distract that grafted tendrimers, then release from that liposomes will happen. That will consist of a tiny nanometer size of that prodrugs will be released from these systems. So these systems will show us 2 points, one area which will enhance the temperatures. That enhancing the temperatures will kill the cancerous cells as well as the cisplatin will also kill that cancerous cells. So we can be able to obtain, or that research group are able to show a 91% suppressing the growth of the tumors when they are using an 41 cells in that animal models. So this is an overall picture which is showing and that earlier systems that initially you will make it as an liposomes by using a fan lipid conjugated with an RGD peptides, okay, dipolymidityl as well as anti-sterile polyethanolamine will make that liposomes. In that liposomes will be extruded into that along with that drug substances, then which will be coated with this this entire thing will be coated with this crafted polymers. So this can be administered into that tumor, that animal having a tumor growth, which will be irradiated using a near NIR light. So once that light is passed, what will happen? There will be an increase in the temperatures that will rupture that cells so that the drug will be get released. That drug will also produce a therapeutic effect, as well as there will be a raise in the temperature that will also induce an cancer cell death. Similarly, I have shown that another case, that is an photothermal hypoxia conditions. I'll come to the pictures now. So as we discuss, once the drug delivery systems administered, which will generate that reactive oxygen species that will induce an hypoxia conditions. At that hypoxia conditions, the systems will be gates breakdown. Okay, that people have used and dialkyl ketyl groups to activate at this stage and to get releases that drug from these systems. So similarly, there is another systems we can talk about it as an photothermal responsive halo nanoparticles, where the system has been developed like an bismuth sulfate halo nanoparticles, which will consist of and are able to provide photothermal as well as some chemotherapy. So that bismuth sulfide nanoparticles along with doxorubicin is loaded in a silicon mesoporous silica, which has been activated due to a light at 808 nanometers. That light activated system will able to generate that heat due to the presence of an bismuth sulfate, which was killed the cancerous cells of an MDA MB213-1. So the temperature was raised up to 49 degrees Celsius under near NIR region, as well as which is sufficient to induces the release of an doxorubicin and ablate that tumorous cells. So this is what that photothermal responsive halo nanoparticles, that is an initially we'll make it as an bismuth that will be put it into a surface of these systems. Then on exposure of a light, which will be releases and generate that heat, both will be responsible for that cancerous cell death. I like to put it up one of an important point. You can see it from a cumulative drug release based on that on-off mechanism. When it is switched is on or light is on, there will be a release of the drug. When it is exposure is stopped, the light or the drug release will not be there. Again, if you are exposed to light, there will be a release. So like that we can expose to a light source, which will be able to alter the drug release at different time points. Based on that, our group is also has done some work in that photodynamic therapy, where we were concentrating on ocular tissues for that treatment of an age-related macular degenerations. As you know that I already mentioned it, that photodynamic therapy will be having a photosensitizer which will be administered into one cancerous cells, which will be placed over there. Once it is observed into the cancerous cells, light will be put it up. That will releases that reactive oxygen species that will kills that cancerous cells. Now, I've shown one case studies before going into our work, wherein we'll be having a true drug of an paclitaxel, which is conjugated with an thiol group, diketyl thiol groups. Okay. Now, the TPC, that is the temperature converting materials. Along with that, we'll be having a paclitaxel prodrugs that both will be put it into an polylactide systems. Okay, so that both has been loaded into an nanoparticles. Okay. PLGA or an polylactide nanoparticles. Now, these nanoparticles has been encapsulated into that RBC membrane. As you know that we'll take that RBC, destroy that internal content, that within this membrane, this nanoparticle has been loaded, that above which that will be targeted for that activation. This system has been administered into that tissues or that cells. Once administered, reaching into that cell membrane, which easily recognized at cancerous tissues due to that targeting moieties present on that surface on activation of that light, which will release or destroy that RBC membrane. The purpose of keeping that RBC membrane to get it as a longer duration of an action. Once it is destroyed, we'll be able to get it as a lot of activation over here. Then similarly, we'll also has that drug over here, paclitaxel, which will be prodrug, which will be on administration. Based on that ROS, which will be gets cleaved and followed by an hydrolysis, that paclitaxel will be get releases. So these stimuli responsive things will happen only within that cancerous cells. So the toxicity produced by the toxol into a normal cells can be prevented. So which has also been shown over here in presence of light and dark systems over here. In case of an dark, there won't be any release of that materials. When you are exposing to that light, there is a degradation of this entire system is happening. So that has been shown very clearly. I already mentioned it, there is a photodynamic that can also be utilized for conversion over here. That is an hypoxia responsive systems, which will activated in hypoxic conditions. Okay. Now, so that for that, they have made the systems which will consist of an doxorubicin, which will be consist of this system has been made up of an conjugated polymers as well as an polyvinyl alcohol. So these two will be put it over here. Within that, we'll be having on that conjugated systems or that conjugated polymer will consist of an two important molecule. One is called as 2-nitroimidazole. Another one is 2-aminoimidazoles. So these amidazole, nitro and iminoimidazoles will, based on that hypoxia conditions, which will alters into that hydrophobic into an hydrophilic conditions. Now, initially, this conjugated polymers has been converted into a nanoparticles by encapsulating and doxorubicin inside. That has been obtained by a double emulsion method. Once that has been done, that light which will be administered into that cells or that patient, which will be activated by light. Once that light is get activated, there will be a dissociation of these polymers, which will also releases that components that further. So the doxorubicin, which is present inside this one, will also released based on that hypoxia conditions, wherein which will be get alters that changes into this hydrophobic into an hydrophilic, that is a sand. Nitroimidazole will be able to convert into an nitroamine. So that due to this reduction, there will be an opening of that system, which will releases the drugs into that system continuously for better therapeutic effects. So now we'll come into our work where that we targeted and developed and photodynamic therapy based systems for treating an age-related macular degenerations. One, in these conditions, in the normal vision, we are able to see clearly. In this AMD, where our vision will be get blurred and leads into that vision loss, which is also treated by a commercially available photodynamic therapy, wherein the drug called vatriforfin will be injected through an IV. After 2 hours, that light will be irradiated onto that eye surface, which will be that photovatriforfin will be gets activated, generate the free radicals that will kills or destroy that newly formed blood vessels. You can see that treatment of that vatriforfin system. Initially, there are a lot of blood vessels. You can see it on that treatment. The newly formed blood vessels are getting decreased. So we also tried for this similar kind of techniques, wherein we have taken up and silver and hypoxia BSS and photosensitizer that both has been loaded into an PLGA, TPGS copolymers, and which is administered into that animals as well as an on light radiation, which will be induces that angiogenesis, anti-angiogenesis effect. What is happening here in case of an hypoxia B on activation of a light, which will be goes into that excited state where that due to an photoactivation, and once it is reaching that excited state due to an intersystem crossing, that triplet state will be converted into one singlet state, during which releases that free radicals or that production of a singlet oxygen. That singlet oxygen induces an apoptosis. In turn, will have that anti-angiogenesis effect, for which we have initially produces an silver, and then we have also produces an PLGA, TPGS random copolymers by ring opening polymerization techniques, and then which has been utilized to make a nanoparticles loaded with an photosensitizer, and that formulation has been optimized by using an design of expert or design of an experiments, wherein we have considered as an two formulation variables and response variables of an entrapment efficiency and particle size has been considered. One is an stirring speed as certain effect on that particle size productions as well as an encapsulation efficiency. So we also consider that polymer concentration into 1:2 ratios and that stirring speed between 700 into 2100 RPM. So within that, we could able to see that loading will be a fun or reduction of the size where the nanoparticles will be with respect to an stirring speed. So then we have started evaluating that formulation by using a chromatographic. We determined the drug content as well as the DSC has been utilized to assess that nature of the drug present in that system. We could find that which is present in an amorphous state, so that we're not able to get it as a short melting point for the drug substances present in that systems. Again, we have evaluated through an by an AFM for the roughness of the systems. So the roughness of the system is important to show enhanced surface plasmon effect. When that light is passed, we are using a silver. That silver will undergoes or produces a surface plasmon effect, and due to which we'll be able to see that more amount of drug release. So that light conversion will be better. So similarly, we have done the other characterizations for that silver and that system. We also studied that drug release for 72 hours, wherein we are able to get it and delayed drug release. So that indicates that at different time points, we can able to make the triggering of release of the drugs. So we found that what could be that optimum concentration of an dose can be given without quenching effect. When you are increasing the concentration of that particles, which will be able to produce an increase in that singlet oxygen productions at a certain point of time, which will also able to produce a quenching effect. Wherein we have shown that photodynamic on-off mechanisms when presence of that light or in an aqueous environment, which is not that much photosensitive, when you are putting a front light, that will able to open up that cells, and so we can clearly see that on-off mechanism is working very well. Now we are also seeing that anti-angiogenic assay by exposing these nanomaterials into initially by a CAMP method, where that egg has been kept taken, where you can see that formation of a new blood vessels. When you are treating with these nanoparticles of this photosensitizer, which is able to destroy that newly formed blood vessels. Again, we have also shown that phototoxicity unto the different concentrations, wherein we'll be able to show that phototoxicity of these materials, and we could also able to optimize the concentration of an photosensitizer, which is used for that formulation, that up to 25 micromolar, which is able to produce an phototoxicity above which is undergoing and quenching, so the toxicity will be get normalized or stabilized. Again, we administered into a rabbit, and we were able to dissect that animals and take the different tissues of that animals and checked about the presence of that components in different tissues. We could able to show that the presence of an photosensitizer in an aqueous humor, retina, iris at different places, which clearly indicates that our IV administered drug substances able to reach that eye and interior parts of an eye, which is also shown that fluorescence of the tissues in retina and iris. You can see it from here, even after 12 hours, that particulate system is present in that iris or within that retina. So we can able to produce that therapeutic effect based on that. That final one, we'll call it as a photoconversion responsive drug delivery systems, where maybe a different mechanism can be utilized, where one is a photolysis, photoreduction, or a photo-switchable strategies. So I have shown one example over here that is based on a photo-switchable strategies, where that upconversion nanoparticles, they have made that upconversion nanoparticles consist of an polyamine amide that has been coated by an DNA polymer, which is conjugated with that azobenzene. So by wearing that azobenzene of a different, that is an DNA polymers of an A, B, and C conjugated with an azobenzene has been made. Okay, now this is that upconversion nanoparticles has been coated with these polymers. Within that, they have also loaded that doxorubicin, that is a drug molecules. Now you can clearly see this is that area where which has been focused over here, that is an yellow or an orange color, where that that azo and cis transform of that azobenzene has been incorporated over here. Within that, the doxorubicin is also loaded. After that, that entire system, after this conversion, that entire system has been put it into an hyaluronic acid. So this hyaluronic acid, so before that, they are also attached an TAT peptide for showing an surface responsive adhesion into the cancerous cells. An entire thing, they put it into an hyaluronic acid hydrogels. Once it goes over there, due to that cancerous cells, that presence of an hyaluronase enzyme, which will destroy that outer areas. Whereas that other cells in the normal cells, which will not be get destroyed. Now, after an specificity, which will be due to that TAT peptide, which will attach into that surface, then internalizations will happen. We'll put it up that light source, so which will switches that cis and trans isomerism, so that will be open, from which the drug will be get releases. So based on this upconversion nanoparticles, also we have done certain work. Shall I take another 5 minutes? Yes, organizer.
You can continue, sir.
Yeah, no problem. So, wherein we have to put an core cell upconversion nanoparticles based on an lantanides, wherein we have considered of taking of an yttrium sodium fluoride as an upconversion nanoparticles. Initially, we have formulated sodium yttrium fluoride as a core material, for which we have taken up an yttrium nitrate in combination with an EDTA and sodium fluoride at different ratios. We have mixed it, and then yes, yeah, I'm coming within 5 minutes. I'm in class. Sorry. So we have done that core nanoparticles by changing that combination of this sodium fluoride as well as an yttrium nitrate, wherein we have kept it as an optimizes that formulation by keeping in different temperature conditions of 200 degrees Celsius, 180 and 160, as well as by changing that sodium fluoride concentration, wherein we will be able to analyze the formed core material belongs to a different morphology as well as an phase. Either it may forms as an cubic or an hexagonal in phase, as well as that morphology wise, it is that produces an sphere, hexagonal rods, or mixture of an hexagonal and spheres. So the prepared core materials has been subjected to an XRD pattern, XRD analysis. From the pattern, we were able to identify that is a molecules were an cubic or an hexagonal or an cubic and hexagonal. That is a way we identified. This is some pictures of that different ratios of that yttrium nitrate and then sodium fluoride. You can able to see that very clear cubic or a sphere-like structure is formed at 200 degrees Celsius. When it is comes into an 180 and an 160, there will be an hexagonal systems also. So when you are increasing that sodium fluoride concentration and decreasing that temperature, you can find it in very clear rod-like structures of the components. This is at 160 degrees Celsius. If you consider, so at 200 degrees Celsius, by varying the concentrations of an sodium fluoride, we are able to get it an sphere as well as an hexagonal rod. When coming into an hexagonal rod, the size is getting increased. Similarly, at 180 and 160, we could be able to find a hexagonal rod as well as an cubic. So based on that, we gone for an what is the efficacy of an photoluminescence, light conversion efficacy. As you know that in upconversion nanoparticles, we irradiate with an near NIR, which will be converted into an near NIR light into an UV visible region. So we have taken that materials and subjected to an photoluminescence, which shows us that mixed phase or that hexagonal phase is showing a better photoluminescence. So we have considered that three phases, both in cubic, hexagonal, and mixed. We could find that hexagonal is showing a better, but in order to keep the size smaller, we sometimes gone for that mixture of that phases also. That next, once we optimize the core, then we also gone for an cell optimization. For cell, we have utilized an sodium samarium fluoride, where we have used that samarium nitrate and then sodium fluoride to make that cell. Similar kind of studies has been carried out by XRD, and we have used as a SIM to identify its morphology and then phase analysis. I would come over here, where also we could able to find it as a spindle-like morphology in all the cases by varying the temperature or by varying the concentration and exposure time. Of every cases, we could able to find it as the formation of an hexagonal, but we want to optimize the size. So at a lower concentration, at 200 degrees Celsius, we were able to produce an hexagonal with an lower size of the particles. So we have prepared and core cell nanoparticles by optimizing the concentrations. I'm not going into much. And then after making a core cell nanoparticles, we have also added a dopant which will further enhances that light photoluminescence activities such as an erbium and an ytterbium. So we can able to, after making that corsal nanoparticles with an dopant, we have done that DLS studies. That core, which consists of 164 nanometers, whereas that core cell will consist of 277 nanometers, and an core cell with an dopant we're able to show and 318 nanometers. So this we have studied by other characterizations also, where we are also subjected to an photoluminescence spectra, where we could find that with dopant or without dopant are able to produce a similar level of an photoluminescence effect. That further, we also subjected that corsal nanoparticles with an HRTIM with an ADAS to identify whether it has all the components. We would be able to confirm that presence of sodium fluoride, samarium, yttrium. So this is an corsal doped, where we'll also have that erbium and an ytterbium. So ER as well as an OR, YB, which is also present over there. So clearly indicates all that components incorporated in that system or that upconversion nanoparticles were present. And for that synthesis of the corsal nanoparticles, we have made it, and then we have coated with that silica layer, for which we have added that silica. Once we have made that silica, then we have made it into a mesoporous. In that, after that mesoporous silica, we have also loaded that drug on it. So these are all the pictures of your corsal silica layer. You can see that is some corsal nanoparticles over which we have produced some silica layer. After making of a silica layer, we have made it into a mesoporous. Due to these mesoporous conditions, we were able to load the drug on it. So this is a sun pattern. You can see that is a sun structure where you will be able to see that porous nature. Again, we also taken that photoluminescence to show that alpha or that mixed phase or that single phase is showing a better efficacy. So we have quantified amount of the drug entrapped, and then we also studied the release pattern, and we could able to show that phototoxicity will be higher on exposure of that light source compared to that without light. So wherein you can see that that material we have exposed into that cell lines, MCF cell line, where in presence of an light source, more amount of an ROS has been generated. That fluorescent image clearly indicates with light irradiation, more amount of an ROS generation. That will be directly correlated for that apodosis or an anti-angiogenic effect of these systems. Yeah, we also done that apoptosis assay, wherein we have shown that is an fluorescent cell-induced apoptosis is by using an anaxin V and a PI staining method, we could able to show that cell death, non-viable apoptotic cells. We can able to see over here. In overall, we can say that again, we have also done that mitochondrial membrane potential, which is also been responsible for that apoptotic activities. In nutshell, I would say that we are able to show the system that upconversion nanoparticles, which is able to produce and apoptotics effect in that cancerous cells. So that further in vivo studies is under process, which will be published very soon. But in the earlier part, we have published in an material research journals. So now I'll come into that conclusion part. Even though I have shown a beautiful part of that a different drug delivery system, which is mediated through that light. But there are a lot of challenges or that critical issues need to be addressed to bring out this system into a market or to a clinical translations. What are all that they? Number one is tissue penetration is less. Tissue penetration of a light source. So that UV visible, which will be less than that one, whereas when you are coming into a near area, we can go up to 5. But still, it need to be improved, or otherwise, this kind of system can be treated only in surface tumor, not inside the tissues or an organs. In order to enhance its capability, that light penetration effect need to be enhanced, or the different upconversion nanoparticulate technology need to be produced. That is that second point, that photon conversion efficiency has to be improved in order to get better therapeutic effect. And that long-term, in vivo long-term biocompatibility and biodegradability of an near IR photoregulative drug delivery systems or any kind of a drug delivery system need to be assessed. Currently, these materials are made up of certain inorganic components or an lanthanide components, as well as some of an organic components also be used, which will be creating a source for the toxicity or that need to be assessed in a long term. That availability of a photoresponsive component is also very less as of now. That photosensitizer, the dyes or the PCM materials are very, very limited. Choices are very limited. That area need to be focused. Due to this limitation, low availability, that manufacturers are low, so the high cost of the production. So you can understand that Hypoxia B, what we have used it for an 50 mg of it is costing more than that 2 lakhs. So that much cost that is involved in this photodynamic therapy or that materials. So that need to be addressed by generating new photosensitizer at a low cost. It is often difficult to reach the tumor region, and that window for this region is also need to be optimized. So these are all critical issues which limits that clinical translations. Why I am putting all those things? This is an important area where that young minds can take up it, the further research and further opportunities for that younger mind to work on it, to bring out a viable light-mediated drug delivery system into that human use. Thank you for your patient listening. Before that, I like to acknowledge some of our collaborators, Dr. Rukmani, Dr. Rajaguru from an BIT campus and University Tiruchirappalli, and my student who has been working on these areas, Dr. Chandrasekhar, Venkateshwaran, and Preeti, who has been contributed to these areas. Thank you for your patient listening. If any questions are there, I'll take it up.
Explaining from chameleon display water bodies to light-mediated targeted delivery system, enlightening us on challenging diseases with novel strategies, Thank you for being an epitome of energy with novel outcomes, sir. And now this session is open for the queries. Good morning, sir.
Yes, ma'am.
Sir, can the light-mediated drug delivery system acquire with the dose control?
Yeah, that depending upon that light source or an excitation of that light, we can able to control that release of the drug material from the system. That need to be optimized. How much amount of that light we will be exposing it, based on that, the dose will be get released.
Morning, sir.
Yes.
Sir, and my question is, what if the drug delivery system releases the drug even before injecting into the body due to the external temperatures?
Yeah, that is what that I have shown in some of that systems. Without that light, the system will not release us that any amount of the drug. Only on exposure of that light, only that polymeric systems or the developed system will release us the drug. That is the advantage. So now we'll make a system, the drug will be encapsulated into that light responsive system. Only on exposure to light only, which will release us the drug. That is that beauty of that systems. There may be the cases, very minor amount of that or minimal amount of the drug may be get released before exposing of the drug. That is the strategy, how you are designing the system. Whether your system is capable to release the drug only on an exposure of that light or in normal also. That efficacy will be depends upon your designing of that system. Either it based on that polymer or based on that covering of that force created in an hollow nanoparticulate systems.
Good morning, sir. I'm Bhima Gulu. Sir, does photodynamic response applicable for light-sensitive drugs?
I'm not getting your point.
Does photodynamic response applicable for light-sensitive drugs?
Okay, that meaning of a light sensitive drug, if it is going to get degraded, then that question will not come over here. That name itself, it is indicating that it is a light sensitive. In what term you will be considering over here? So I can say that it is the photosensitizer is also light sensitive. Okay, how you are considering it? If you are talking about the stability point of view, that is a different scenario. Here we will be considering as it is a sensitive to light. Then only which will be able to convert or release or excite from a ground state into an excited state. If it is not a sensitive, it's not going to happen anything.
Okay.
But you have to think I think hopefully you might be expecting answers related to the stability of that material. If I say that once the light is exposed to that material, if it gets degraded, originality will be get lost, then that cannot be utilized.
Thank you.
Good morning, sir. This is Jayashree from Quality Assurance Department. Sir, this is Jayashree from Quality Assurance Department. Sir, my question is, what is the reason to use laser light at 590 nanometers?
Can you repeat it? Your
What
voice.
What is the reason to use laser light at 590 nanometers?
I'm not getting your voice clear. You keep it somewhat out because it's echoing.
So my question is, what is the reason to use laser light at 590 nanometers?
Oh. So that is the basic concept of the study. We have to use that laser lights, which will be having a different wavelength. I have shown you that initially itself, if that wavelength is shorter, that will be having a high energy, that you know that, but the penetration effect across the skin is low. So we'll be going for a different region of that laser sources or a laser light. I mean that region of near IR will be using it nowadays for that photo conversion or up conversion technologies. So in order to generate that light source, we'll be using that lasers. Hope I answered your questions.
Yes, sir.
Yeah, thank you.
Now, as a token of appreciation, please accept the e-certificate on behalf of Sri Vishnu College of Pharmacy. This will be sent by your mail, sir. Please kindly accept it.
Yeah, thank you, madam.
Thank you, sir. Now I request Dr. Ashok Tuluru, Organizing Secretary, to conclude the session.
Yeah, very grand morning to the guest speaker as well as all the delegates. And it is the overwhelming performance by Professor Subramanian Nateshan, sir, Department of Pharmaceutics, NIPER, Kolkata. Thank you, sir, for enlightening all our speakers on the new concept and a novel concept of light-mediated drug delivery system as a part of the targeted drug, as a part of the theme, our targeted drug delivery system. And we are also very pleased to inform you, we are having the registrations of around 1,500 participants from 23 different states and 2 union territories. And we are giving it live streaming through YouTube. Most of the other participants also, they are requesting you to share this presentation, sir. With your permission, can we have this PPT to be shared with them? If there is no problem, can we share it with the remaining participants?
Sir, I think I have told some of the results are unpublished results. I told that too.
Okay, sir. It is coming.
Yeah, because I want to give the latest trend so that I have given them some of that details. Most of them is okay, but some of that back part it is having an unpublished data.
Okay. Okay, sir. In that case, they will have the data from the YouTube itself, the recorded version. And I have a small announcement to all the participants. Every day we are giving the attendance evaluation and feedback form through your registered emails as well as the groups of WhatsApp groups in which you have been registered. Please, everyone has to fill them and submit the 3 days feedback form. Then only you will be eligible to get the participation certificate. Thank you to all, and let us meet tomorrow and the concluding session of this 3-day national webinar. Thank you to one and all.
Thank you, sir. Thank you.